Good afternoon and welcome virtually to Old Dominion University and its remote experience for young engineers and scientists program known as REYES. My name is Giovanna Gennard, Assistant Vice President for Strategic Communication and Marketing and co-chair of REYES, and I will serve as your host and moderator for today's session on past, present, and future of Florida's coral reef featuring Haley Burleson. The purpose of the Global REYES program is to offer free virtual learning experiences that increase science literacy, inspire and train future generations of STEM-age students. And today's discussion with Haley Burleson offers us insight into the current environmental stressors Florida's coral reef is experiencing and what Mote Marine Lab's International Center for Coral Reef Research and Restoration is doing to help prevent it from going extinct. Throughout the session, you may submit questions through the Q&A box on your Zoom screen. And following the presentation, we will answer as many of your questions as time allows. It is now my pleasure to introduce our guest speaker, Haley Burleson. Haley, would you please turn on your microphone and video so we may see you?
Hi, everyone. Thank
you.
Hi.
Haley Burleson is the Community Events Coordinator at Mote's Marine Laboratory and Aquarium's International Center for Coral Reef Research and Restoration on Summerlin Key in the Florida Keys. We are very proud of the fact that she earned her undergraduate degree in marine biology from Old Dominion University, where she was also the star of many of our marketing campaigns, highlighting her amazing accomplishments in research and her innovative work. She also recently completed a graduate certificate in nonprofit leadership through the University of Florida. Haley has an extensive background in fisheries research, which is how she began working with Mote Marine Laboratory and Aquarium as a fisheries research technician. She ultimately made the decision to leave the research field to be in a position that would help raise awareness of local environmental issues while simultaneously putting the scientists and their research into the public eye, all in an effort to think globally and act locally as a community. She's responsible for supporting the communications and development activities for Mote in the Florida Keys and the institutional goals of promoting Mote's research, generating attendance for their public exhibits, education programs and events, and also supporting fundraising efforts while also enhancing the institution's reputation, integrity, and partnerships. Haley also manages the social media pages for Mote's International Center for Coral Reef Research and Restoration. So if you want to follow them, be sure to follow at Protect Our Reefs. In addition to working with Mote Marine Laboratory and Aquarium, Haley also volunteers as secretary for the Florida Keys chapter of the Surf Rider Foundation, and she is a member of her local Toastmasters organization. Please join me in welcoming our very own Haley Burleson to REYES. Welcome, Haley.
Hi, Giovanna. Thank you for having me. I am excited to be here, a little nervous, but excited. So yeah, I work at Mote's International Center for Coral Reef Research and Restoration, and I know that's a mouthful, so I will be referring to that as IC2R3, which is our acronym. I'm going to share my screen and get my presentation together for you guys. Give me 1 second. Alrighty, can everyone see my screen?
We can, yes.
Perfect. So good afternoon, good morning. My name is Haley Burleson. Like Giovanna said, I am the Community Events Coordinator for Mote Marine Laboratory's International Center for Coral Reef Research and Restoration, otherwise known as IC2R3. I graduated from Old Dominion University with a bachelor's in marine biology. And like Giovanna said, you probably have seen my face on some advertisement around the local Norfolk, Virginia Beach area. I'm going to talk to you today about the past, present, and future of Florida's coral reef. But before I do that, I need to give you a little bit of a background of who Mote is. So let's get started. All right. So this woman right here is Dr. Eugenie Clark, and she is a famous shark scientist. She is more popularly known as the Shark Lady. And in 1954, she published her book called Lady with a Spear, which is her memoir of her research that she did in the Red Sea. This is the book that grabbed the attention of the Vanderbilts, who ultimately gave her funding to have her own laboratory in Cape Hayes, or in Placida, Florida. And this laboratory was called the Cape Hayes Marine Laboratory, where she did her shark research. She invited other researchers from around the world to help her on her shark research. And this is her doing some behavioral studies in one of her shark pens at the Cape Hayes Marine Laboratory. By 1960, it was clear that she was growing so much that she was going to need a new space. So the Sarasota County and the city of Sarasota wanted to have a research facility in their city. And they saw that she needed more space, so they gave her this new location on Siesta Key. But by 1967, the lab was named to honor a major benefactor, also known as William Mote, which is how we got our name today as Mote Marine Laboratory. So Mote is after a person, and it's not an acronym, and a lot of people think it's an acronym. So it's actually after a major benefactor known as Mote. Mr. Mote had recruited Dr. Perry Gilbert to transform the lab from a field station to a major research center. In turn, Dr. Gilbert expanded Mote's research programs into 5 different fields: microbiology, neurobiology and behavior, estuarine ecology, the biology of sharks, and biomedical studies. This photo is of scientists loading a basking shark onto a boat for research. In 1976, the lease on Siesta Key expired and Mote Marine Lab moved to where it remains today on City Island in Sarasota. At this location, Mote now has over 20 different research departments, and we also have an aquarium that highlights our research. It's at this location that we do most of our coral health and disease research because we don't want to cross-contaminate anything in our Florida Keys lab. But to enter the national arena of coral reef research, Mote had to first secure a lab space in the Florida Keys, where the Florida Keys Reef is. In 1993, Mote established their presence in the Keys by partnering with the Pigeon Key Foundation. And this is Pigeon Key here, and this location is where Mote had their first coral laboratory. But in 1998, Hurricane George came through and pretty much destroyed the property. So in 2000, Mote secured a new location on Summerlin Key, which is where we remain today. We first started as a small building, this one white one to the left that you see here. But in 2016, it was clear that we were going to need way more space because we had grown out of that little building. So we demolished it and we built the massive building that we have today. This is a 19,000 square foot laboratory that has dormitories for visiting scientists, 2 wet labs, 4 dry labs. The whole property is filled with raceways that hold coral. It's a beast of a lab. It's category 5 rated. We have solar panels on the top and we have cisterns, and these solar panels and these cisterns help us to be a sustainable building, which gives us the certification of a LEED Gold certified building. LEED is an acronym for Leadership in Energy and Environmental Design. And this is essentially a green building, and it's the first of its kind in the Florida Keys. The solar panels provide up to 18% of our energy, and the cisterns collect rainwater that we use in our restrooms. You can see on the backside of the property here that at the beginning, when it was first built, we had a couple of raceways, which are those blue rectangles that you see. We now have 80 raceways on the property, and each raceway can hold up to 1,000 coral fragments. It is here at this building, the International Center for Coral Reef Research and Restoration, that Mote scientists conduct high quality research on coral health and disease, coral restoration, coral resilience, coral reproduction, coral reef monitoring and assessment, and also ocean acidification. So now that everyone has had a little crash course of Mote, I think it's time for a little bit of a breather. So what I want you all to do with me is just take one big deep breath in and just let it out. And I want you to ask yourself where that oxygen just came from. And if you said trees, well, I hate to break it to you, but you're wrong. We also have, well, let me backtrack. We also have an underwater nursery, but where the oxygen that you just breathed in comes from the ocean. And the scientists estimate that 50 to 80% of the oxygen that we breathe comes from the ocean, and which is why it's so important to protect our ocean ecosystems like coral reefs. But before I want to get into coral reefs, I want to make sure that everyone knows that corals are animals, and they're not like plants or rocks that some people might think they are. They have a calcium carbonate skeleton, and they have a cup-shaped skeleton that's made of corallite. Each coral has a stomach and a mouth, and they have these tentacles that have nematocysts in them, and those nematocysts are stinging cells, and they can use those stinging cells to grab any small fish or drifting plankton that come by and feed them. But they also have zooxanthellae in their coral tissue. And so this zooxanthellae is the brown spots that you see here. And the zooxanthellae has a symbiotic relationship with the coral. The coral tissue is actually translucent, and the zooxanthellae is what gives the coral its color. And when I say symbiotic relationship, this really just means that the coral helps the zooxanthellae, and the zooxanthellae helps the coral. So corals actually get about 90% of their energy from the photosynthesis that the zooxanthellae creates. And so when the zooxanthellae photosynthesizes, it releases the energy products that the coral will take in, and then the coral will release waste products that the zooxanthellae will eventually take in and just continue this cycle. So essentially, coral tissue is clear, and zooxanthellae gives it its color, and it feeds the coral. This is a really important relationship that corals have. So now we're talking about Florida's coral reef, right? Like, this is the red area of Florida's coral reef. It stretches all the way from Martin County all the way to the Dry Tortugas, and that's about 350 miles of reef that we have. But why is the reef so important, right? We know that it gives us, like, the ocean gives us oxygen, and like corals are animals, that's so cool. But coral reefs support up to 25% of marine life, like these tarpon you see here. And this is one of the reasons why Florida is the fishing capital of the United States. They provide structure, habitat, bait, and hunting and breeding grounds for many of Florida's saltwater food and game fish. Coral reefs also provide protection from storm surge, from storms like hurricanes and tropical storms. And in 2016, when Irma hit the Keys, Lou Key, where that red pin is at the bottom of your screen, saw 20 to 25 foot waves, whereas our lab on Summerland, which is on the other end of this yellow line, saw 4 to 5 foot waves. So that's about an 8.5 mile distance that saved us about 15 to 20 feet of storm surge. We still had damage come through when Hurricane Irma hit, but it could have been a lot worse if the reef wasn't there to protect us. The Florida Keys welcomes millions of visitors every year, and it also is responsible for providing up to $2.4 billion annually to Florida's economy. So this is another reason why it's really important to protect our reefs, because the reef helps us to make a living. The reef also provides up to 70,000 jobs for us down here. And this is something that I really want to tell you guys that's so important to us, is because this reef is what we make our livelihoods off of. We live on the reef, and we play on the reef, and we work on the reef, and this reef is really our home. and we all rely on it in one way or another. You could be a fishing guide that guides all day and earns your living from guiding fishermen out to their sport fish. And you can also collect your food from being out there to feed your families. You could be an ecotourism boat who relies on tourism to fund your business by taking all the tourists out. Or you could be a small business owner that relies on tourists and locals alike to buy your products. You know, the list goes on. And like I said, it's our livelihood down here, this beautiful, delicate ecosystem. We live here, we work here, and we play here. Like, this is our home. And it's important for us to protect it. And so when I say that my job is to protect our reefs by spreading the awareness of coral reef ecosystems, I'm doing much more than just trying to protect a delicate ecosystem. I'm also trying to make sure that the reef stays here to protect the livelihoods of all of the locals who live in the Florida Keys and that rely on this delicate ecosystem to fund their businesses and feed their families. So this is what one of the reefs looked like in the Florida Keys in 1975. And you can see here there's an abundance of elkhorn coral and reef fish. But unfortunately, since the 1970s, there has been an estimated decrease of 90% of our indigenous coral coverage, including some of the species that has experienced a decline of more than 97%. This is the same location, but just a decade later in 1985, and in 2004, and in 2014. Just imagine what it is today in 2021. This coral degradation is what's occurring throughout Florida's coral reef. A very noticeable difference, right? So to be called a healthy reef, there has to be a minimum of 24% coral tissue cover. And currently, Florida's coral reef has less than 5% coral tissue cover. And with this minimal tissue cover, it makes wild coral spawning nearly impossible without human intervention. And because of this, scientists are calling Florida's coral reef functionally extinct. So how did we get here? Well, years of pollution, disease outbreaks, habitat loss, physical impacts, and warming waters. But one of the final nails in the coffin was when the stony coral tissue loss disease came about. In 2014, in Miami-Dade County, a new disease outbreak was affecting the stony corals. And stony corals are one of those major reef building corals. So this is really important to save these corals from this disease. However, scientists don't know if the disease is bacterial or it's viral. In a lab setting, it's shown response to a bacterial ointment or antibiotic ointment. So there's some promise there, but research still needs to be done to really confirm what the cause of this disease outbreak is. So stony coral tissue loss disease started in 2014, and it's moved up and down the Keys all the way down to the dry Tortugas. And unfortunately, last month, when scientists were conducting a routine coral survey, they came across this elliptical star coral that had the stony coral tissue loss disease. And they acted quickly to provide a treatment on the coral, which you see here. This paste is what we use. It is a mix of amoxicillin or some sort of antibiotic and another compound that scientists have stirred up together to make this paste, and it provides a temporary treatment. And they apply this along the disease line in an effort to keep the disease from spreading across the whole coral. And so when the stony coral tissue loss disease affects a coral, it essentially just peels off of their tissue, and you're just left with this stark white skeleton. But it's kind of deceiving because it also looks like coral bleaching. These are pillar corals that have bleached, and if I go back a slide, you can see how it looks similar. But the difference between bleaching is bleaching occurs when waters warm to a point to where when their zooxanthellae get too stressed out and they leave. And like I said, coral tissue is translucent, so of course you can just see right through to the skeleton. Now, the zooxanthellae can come back if waters cool down a little bit, and the corals will regain their zooxanthellae and their color and keep getting that energy. But if waters stay warm for too long and the zooxanthellae has left for good, then the corals are not getting that energy that they need, and then they eventually die. So it probably sounds like I'm the bearer of bad news right now. I'm sitting here telling you like a whole ecosystem is about to go extinct, and that's not good, right? But I do have some good news. Remember how I said that we have scientists that study a plethora of topics in coral reef research? Well, they do some pretty cool stuff, and I'm about to walk you through the cycle of coral restoration. This is one of our scientists outplanting staghorn coral fragments. Staghorn is a type of branching coral, so they are not affected by the stony coral tissue loss disease, but they do have their own sort of diseases that they get occasionally. And just like humans do, we all get diseases every now and then, and we also have treatments for them. So it's just the natural cycle of the environment. If an animal gets a disease, we treat it. But how we outplant our staghorn corals is we take a railroad nail and we hammer that into the hard substrate of a dead coral head, and then we take a zip tie and we wrap that around our branching coral fragment and we tie it tight. And then over the course of the years, they'll just branch out and keep growing, and they'll secure themselves to the substrate, and then they'll grow thicker, and then you'll have this big thicket of staghorn branching corals on the reef. So this is the coral reef restoration cycle, and Moat can do all of this in-house. We start with spawning, and then we raise our corals up, and then we microfragment them, which is a form of asexual reproduction. We put them through our resilience trials, and then we outplant them onto the reef to where over the course of 5 to 10 years, we're hopeful that they will grow and fuse and be big enough to spawn on their own. And like I said, corals can reproduce asexually and sexually, and I'm about to walk you through that, and we're going to start with the sexual reproduction cycle first. This is what it looks like when corals are setting. And what I mean by that is if you see these spheres in the middle of each coral polyp, that is a gamete bundle. And these gamete bundles are about to be released into the water column, where they will then mix and fertilize and then settle back down to the reef. So corals spawn once a year. It's around the first full moon in August. And remember how I said we have less than 5% coral tissue cover on the reef? So that makes it really hard for wild coral spawning, right? So if these corals are spawning and releasing their gamete bundles, they're not meeting other coral colonies to mix and fertilize and settle back down. So it's pretty much just like a wasted spawn. And that's where we come in, because we provide the human interaction for that. This is one of our adult spawning staghorn colonies in our Lukey nursery. We have this colony on a coral tree out there, and every summer we will go and collect our adult colonies of staghorn and elkhorn coral. And this is what the collection process looks like. We have a team of divers, and we have a bunch of baskets. We have topside support, and we go down underwater to our coral tree, and we make sure that all of it is in alignment. So we find the genotype of coral that we're collecting, and we'll bring it down to our basket that has the same label for that genotype, so we don't get anything mixed around. And then once we have them in our baskets, we also collect our elkhorn coral. These are all of our baskets that have the staghorn coral in them, and we will bring them back up to the boat. It's a big workout. And we pass them off topside. And then our topside crew will take the corals out and put them in these totes that we have filled up with water. And then from there, we will take them back to the lab, where we have them separated in these large coral tanks. So you can see the barrier in the middle and the multiple staghorn coral colonies on each side. This is how we can tell the genotypes apart. We have like genotype 7 and 11 and all of these different numbers throughout our spawning system. It's important that we know the different genotypes, because we have genotypes that have been more resilient to coral bleaching and more resilient to acidification and more resilient to disease. And so when we're spawning our new corals, we want to make these cross fertilizations of like, maybe we want a coral that has a high resilience to coral bleaching, or maybe we want a coral that has a high resilience to disease. And so that's really why it's important for us to know what coral genotypes we are collecting. And so here we move on to collecting, we move on to fertilizing these eggs. So this is our cornflake stage. And those rigid cornflake looking things are actually fertilized eggs. And the perfectly spherical one is an unfertilized egg. So when those gamete bundles release into the water column, we'll collect them with these little pipettes, and then we'll put them in a falcon tube, and then our scientists take them up to the lab where they will then separate the genotypes and then mix them and fertilize them. And this is how they know how successful the fertilization was. So this fertilization was about 90–95% successful, because we only have one unfertilized egg here. And it's important, again, to know the genotypes and fertilization rates because of resiliency. We want those resilient corals. And so with all of those diseases and climate changes that corals are facing, we can't outplant, and we don't want to outplant corals that are just going to die from the first stressor that they see. So again, this is why resilience is super important to us. We want to outplant corals that are going to be strong enough to withstand current and future ocean conditions. And we can know which genotypes are favorable through the use of our chaos system. Our chaos system is an acronym for our Climate and Acidification Ocean Simulator. And this is a beast of a system that can manipulate current and future ocean conditions. It's here where we can determine which genotypes have certain resilience properties. And some of these tanks might have a higher pH, and some of these tanks might have a higher temperature. And so this is pretty much a stress test for our corals. And this is how we know which ones are going to be strong enough to outplant back onto the reef. And because of our science-based restoration techniques, we have more than 95% survival rate after 1 year. And a really great example of this is when we have our Newfound Harbor outplants. So this is a colony that was outplanted in 2015. And these corals have survived a record bleaching event, Hurricane Irma, and the stony coral tissue loss disease outbreak. And last year in 2020, Moat scientists had a breakthrough in coral restoration. And so for the first time ever, restored corals to Florida's coral reef spawned, and we caught it all on camera from beginning to end, which closed the loop on coral restoration. It was at this moment here in this photo that Dr. Hannah Cook, our coral reproduction biologist, discovered that our outplants from 2015 were gravid, meaning that they were pregnant, and they were ready to spawn. So take a look at how large this coral colony is, and like keep that in your brain for later when I talk about how we outplant corals. So how did we know that the corals were gravid? So what we do is we take a core sample from the colony and we look really closely at it. And if you look really closely, you can see that little white squiggle and little pink round things. Those are the sperm and the eggs. And together, that's the gamete bundle, which was in the mouth of the polyp at the beginning of this spawning process that I showed you. So then every night during the spawning window, our scientists went out to the reef and stayed underwater until they saw some magic happening. And they saw the coral setting, which is this. Again, the gamete bundles are in the mouth of the polyp, and noting that they're setting and they're getting ready to release them. And shortly after, they spawned, and we caught it all on camera, and it was so good. And I'm going to try to show you a video, and I don't think it's the best quality, but we're going to try to do it anyway. So you can see how they floated up from the mouth of the polyp into the water column and just swayed back and forth and let the current take them. So we didn't collect those gamete bundles because the purpose of that was just to make sure that they spawned. But at the lab, we do collect the gamete bundles. And this is a gamete bundle floating up from the mouth of a coral polyp. And like I said, we take little pipettes and we suck them up and we put them into these falcon tubes. And then we take them back to the lab to get fertilized. And once they're fertilized and they settle on our coral plugs, we have coral babies. So if you can see these dark brown spots on the purple circles, which are called plugs, those little dark brown spots are coral babies. And the reason why these plugs are purple is because we sprinkle this stuff called crustose coralline algae on the plugs, which is, the acronym is CCA. So if you hear me referring to it as CCA, that's what I'm talking about. But the CCA is an attractant for this coral larva to settle on the substrate. Essentially, it mimics the environmental cue that they're looking for to settle onto the reef. And this is how we are able to get them to pick a spot on our coral plugs to settle and then grow. And once they grow a little bit bigger, they'll go out to our land-based nursery. And you can see here that they are a little bit bigger and the coral plugs are still purple, but you can see that the brown spots have gotten a little bit bigger. So they'll go here to our land-based nursery and we'll have our scientists take care of them until they fill the whole size of the plug. And each plug is about the size of a quarter. And then once they fill the whole size of the plug, we take them into our asexual reproduction process. So they'll look like this, full size, about the size of a quarter, and then we'll microfragment them with a diamond blade saw. And when we do this, we're cutting them into 1 centimeter squares. And then we'll find a plug, and these plugs have their genotype and their scientific name on them. So the OF here that you see stands for Orbicella fabulata, which is the great star coral, and 11 is the genotype number. So this is Orbicella fabulata genotype 11. And this is important, again, to note which genotype we are outplanting onto the reef, and I'll get to that in a little bit more detail later. But once we have them on our coral plugs, we'll take marine super glue and we'll glue them to the plug, and we do this over and over again until we have a lot of these fragments. And once these fragments go fill up their plug again, we take them back to the reef and we outplant them onto a dead coral head with the hope that within 5 to 10 years, they will grow over the coral head, fuse together, and become reproductive size. And here is a video of the process. We take a power drill and drill holes into a dead coral head, take some marine epoxy, mold it up, and then we'll take some coral plugs and we'll wrap the epoxy around the coral plug. And then we'll put them in the holes we just drilled. And we do this in like a pepperoni pizza style pattern because we want them close enough that they'll fuse together within a short time period. And that's what it looks like at the end. And so we do that with the hope of that they will grow to be this size, which is a reproductive size. This is the same colony that Dr. Hannah Cook found was gravid. This is the one that we documented spawning in the wild. And so when we outplant them in the pepperoni pizza style pattern, again, we want to plant them close enough that they'll fuse together over the course of 5 to 10 years so that they become a large colony like this one. And it's important because corals reproduce based off of size and not age. And so with minimal coral tissue cover on the reef, it's important that we reskin the reef as quickly as we can so they can reproduce on their own as quickly as they can, or as soon as they can. And so in the wild, it takes about a coral to grow this size in about 50 to 100 years. And we've managed to speed up the process to get them to be reproductive size in about 5 to 10 years. So we've really sped up the process, and it's amazing work that our scientists are doing. But have you noticed that we've come full circle now? We started with spawning, we've raised them, we've asexually reproduced them by microfragging them, and then we've gone and outplanted them, and then they've grown large enough that they've spawned again on their own. But there's more work that needs to be done, right? Like this is just a Band-Aid that we're doing. It's a stopgap. It's not really fixing the problem because there's still climate change that's occurring. And so Moat recently built a coral gene bank at their aquaculture park in Sarasota because we want to plan for worst case scenario. What if a genotype goes extinct? Well, now we have genotypes at our lab in Sarasota that we can use to reproduce and do studies on for future research. And we'll always have those genotypes saved. And so it's a safety net for us, in other words. So what's the future of coral reefs, right? Well, NOAA, the National Ocean and Atmospheric Administration, recently implemented their 7 Iconic Reefs mission, which is a $10 million mission to restore these 7 iconic reefs in the Florida Keys. And Moat is one of the partners on this project. 7 Iconic Reefs initiative uses a phased implementation strategy focusing on genetic diversity and resiliency to restore multiple foundational coral species in multiple habitat locations across 7 iconic reefs, all with the hope to restore Florida's coral reef back to 15% coral tissue cover in 10 years and eventually 25% coral tissue cover in the next 20 years, which would bring back the reef to a healthy reef status. And so to wrap up everything, this is a short video that pretty much puts everything into a nutshell. So enjoy. And with that, I will take any questions. Let me know if I need to stop sharing my screen.
Yes, if you don't mind stop sharing your screen, then we can go into the Q&A session. And to our audience, I invite you to submit questions, and we promise to answer live as many as time allows. So thank you again, Haley, for talking with us about the science behind the Florida coral reef restoration efforts that are happening in the Florida Keys. And so we do have one question, and that is, what part of the job do you work on?
So my role at Moat is the community events coordinator. So I really lead most of the outreach programs that we have, any events in the Florida Keys. I also work on our partnership development, and I also work on marketing our Protect Our Reefs license plate. The state of Florida has a specialty license plate that is called the Protect Our Reefs license plate, and it's this pretty reef scene, and $25 from each purchase comes back to help fund our laboratory and our coral reef research. And some of that money also goes into a grant fund for other scientists that are studying coral reefs. They can apply for a grant, and they can use that money to do their own coral reef research as well. So I'm pretty dynamic in the world of communications and marketing. That's what I do.
And then can you tell us a little bit about before work? Because I know that you have conducted research, and I remember you having internships while you were at ODU. Can you talk a little bit about your experience there and what took you to the Moat?
Yeah, so I got my undergraduate at ODU, and I was studying marine biology. And as I was wrapping up the end of my undergraduate career, I was working as a research technician at the Virginia Institute of Marine Science in their fisheries research program. And that's really where I gained a lot of my fisheries research. And so once I graduated ODU, I bounced around doing some seasonal fisheries jobs, and I eventually landed a full-time research technician job with the Snook Stock Enhancement Program at Mote Marine Lab. And that was how I got in with Mote Marine Lab, was as a fisheries research technician. And then after being there for a little over a year, I just started to notice how people always were wondering what we were doing with the research. Because the sampling that we did was essentially in people's backyards. We did it up creeks and little estuaries. So we would launch out of people's backyards sometimes, or we would pull nets in people's backyards because they lived right along the creek. And people would come out and they'd ask like what we were doing. And I'm just like, how do you not know what we're doing? This is literally your backyard. And so that's kind of what changed my brain to realize that there is this large gap in communicating science to the general public. And I wanted to be more in a position that bridged that gap, because I think that it's really important that people are aware of what's happening in their local community, whether that's in on the ground, on the surface, or underwater. So I just chose the underwater way of trying to spread awareness of what's going on in the community.
Well, I know certainly today you taught us so much, including the fact that the majority of the air that we breathe is actually thanks to the ocean and not necessarily trees, which is what we've been taught all of our lives. And so that was really impactful, just learning that bit of data. We have some more questions from our audience, and one of them is, knowing that you're the events coordinator, how often do you get to dive? Is the goal to continue to save, protect, and help grow the coral reefs? Excellent question.
Yeah, that's a great question. So I don't get to dive all that much because that's not my job. We actually have a very high standard and quality of divers that we have as our staff. And I'm not qualified enough to dive with our staff. I just have the basic open water certification. I am currently working on my advanced open water certification, but all of our divers have the research diver certification, and they have hundreds, if not thousands, of dives under their belts. And I only have like 10. So
you'll get there.
Yeah. So I'm not near qualified enough to dive with them. However, I do dive recreationally, and I do like doing that. I do get to go on boats sometimes. We have a program with our local dive shop down here. They are Captain Hooks. They're on Big Pine. And every Tuesday we have a moat scientist on the board, or on topside on the boat. And they're there to talk to the tourists that are diving the reef about coral reefs and coral reef restoration, and just, be interactive with the public in that sense. The second part of that question was how long I plan, or what's the long-term goal here? Is that what the second part of that question was?
Yes.
I mean, so our coral restoration scientists always joke that they would like to be out of a job, right? Because as long as the coral reef is not healthy, they have a job. But ideally, we want the coral reef to be healthy, which would in turn put us out of a job. So long-term goal is to put ourselves out of a job, essentially. We want to be able to reskin the reef so it's healthy, and then maintain that. But again, there's a lot of pressure on the reef, from climate change to just human interaction. There's thousands of boats that go to the reef to put tourists on there to show them around. And that's emissions from boats. It's like storms come through and they knock the coral off of their structure. And there's also accidents like runagrounds and anchors. There's a lot of things that are happening. So it's important for us to continue to educate people on how to use the best practices to keep everything safe underwater.
Absolutely. And speaking of best practices, what are some ways where we can make a difference when we visit the oceans, right? Mm-hmm. So for example, I've noticed that some of the new sunscreens tell you to please be careful in the type that you wear, because it could actually impact coral reefs. Can you talk to us a little bit more about what are some ways that we can help protect the coral reefs?
Yeah. So with the sunscreen, there still needs to be more research done on it, because there's a couple components that are not healthy for the reef. But again, more research will confirm that. The best sunscreen to use is something that's more of a mineral sunscreen. And then what we always tell people too is just to cover up. If you ever come down to Florida, especially the Florida Keys, it's hot down here, and you'll notice that there's a lot of people that are covered head to toe, like in the photo that had the flat sky. He has a buff over his face, a hat. He's got a UPF shirt on. It's long sleeve, and he's got pants on, and he probably has flip-flops on, but that's about it. We all just kind of cover ourselves because that's the best way that we know to protect it, is just to not even put anything on. And a side note to all of that, our scientists don't wear sunscreen. They don't put perfume on. They don't put lotions on when they're working in our tanks, because we don't want anything to contaminate the corals in the tanks either. So we're all very natural here. But in regards to traveling to the Florida Keys and when you're around an ocean ecosystem, the best practice to use is what I say is know before you go. Do your local research and the issues that's in that environment. And there are more than likely organizations that you can link up with that will let you do some sort of like cleanup or educate you on what you're looking at or what you're visiting. So down here, like I said, we have our Captain Hook's program. It's called Moat on the Boat. And we put a scientist on the boat to educate people. And other than just like taking your trash with you, don't pollute, don't throw things in the ocean. Use reusables, make a minimal impact in your own life. A lot of people down here have reusable water bottles, and they have reusable straws. There is even a local organization down in Key West who has a Skip the Straw campaign, and a lot of the restaurants in Key West, and it's even brought up the Keys a little bit, has vowed to skip the straw, and they will not serve you a plastic straw at their restaurant. And even if you ask for one, they won't give you one because they don't have them. So just little things like that, small things daily will make a big impact.
That's really important to note. And in fact, that campaign of skipping the straw is coming over to Virginia, because I noticed at Old Dominion, we're located in Virginia in the US, and we're actually a waterfront campus, right? So we're right on the coast of the East Coast of the United States. And in many restaurants locally, I've noticed that they no longer give you a straw, and they ask that you not request one either, because they want to make sure that we're, following this effort to continue to protect our oceans. Mm-hmm. So it's really great to hear.
Yeah, I mean, and it's more than just plastic straws too. It's plastic bags, it's plastic bottles, it's even those plastic rings that you get on your soda cans. And now that COVID hit, those single-use masks are a problem too. So if you have to dispose of a mask, cut it up so it doesn't get wrapped around anything. If you have to dispose of anything like the plastic rings, I cut those up as well. Yeah, it's crazy. I would love to have a limit on plastic bags, but that's a dream.
I know some states are actually already starting to impose them, so hopefully that will be in the future. Mm-hmm. And I mean, all these things go to show to our audience that we can all be environmentalists, right? We can all take time to make daily changes so that we can help protect the environment in which we live. And we have another really great question. This is from Clark Q. And this person is asking, first they say, great job, Haley. Could you please elaborate how the Genome Library in Sarasota safely stores the various genomes? Are they living, frozen, or what?
So we have both. We do cryopreservation of our genotypes, and then we also have them live in the raceways. And I'm fairly certain we do cryopreservation because I've seen them do it. But it's essentially the same system that we have at the lab in the Florida Keys, with the blue raceway, and the coral plugs are on that white rack. And we have scientists up there maintaining those raceways, and it's just kind of a living gene bank is essentially what it is. So they're stored safely up there. We have staff to monitor it, and we have all of the alarm systems in place. So if something does go wrong, we are alerted and can fix it as quickly as possible.
Yeah, it's incredible to see all the work that takes place in protecting them, and even things like a nursery, right? You're talking about all the precautions that are taken when you go in the nursery, just like you would when you go in a baby nursery in a hospital, right? Mm-hmm. people are wearing their masks and their gloves and handling babies with great care. And you do the same thing with the corals. So that's great to hear. And then this person would like to know how long you may plan on working for your current company and continuing to do the work that you do in Key West.
Honestly, I don't see an end in sight. I got into the marine biology field because I wanted to make a difference in the environment. And I have truly found that to be a very real thing for me down here. There is a great need to protect our reefs, and I have a great job where I get to talk to people about the importance of the reef and how cool it is. And I've learned so much just from being in this position, but I also know that I have a lot more to learn. So I don't see myself going anywhere anytime soon. I really enjoy what I do.
And what opportunities are there for volunteering at the moat or internships?
So we do internships. Every summer we have a lot of interns. You would just have to go onto our moat website, which is moat.org, and there will be some sort of tab at the top or to the side of employment opportunities or internships and volunteering. Our Sarasota campus uses volunteers a lot. However, since our Florida Keys campus is so new to being large enough that we can have volunteers, we're still trying to figure out that process. And then when COVID hit, it kind of threw us for a loop. So we are still in the beginning stages of figuring out how we can utilize volunteers at our Florida Keys campus.
And then we have a question from actually a master naturalist from Pennsylvania, Beth Clark. She is planning on visiting the Keys and wanted to see if snorkeling provides visitors with enough of an experience, or is scuba really the way to go when wanting to learn more about the Florida Reef?
So snorkeling will do what you want it to do. The Lou Key, the one that's popular in the lower keys down here for us, that reef is about 25 feet underwater. So even with snorkeling, you'll be able to see it because the visibility is clear as day. I got certified in scuba diving in a quarry in Northern Virginia, and the visibility was horrible. So when the visibility is horrible down here, it's still a clear day to me because I'm not used to it being this clear. So snorkeling you will see enough from snorkeling. The only reason you would want to scuba dive is to just kind of like get down and under and see everything a little closer up. You could see eels or lobsters or things like that that are more on the bottom of the reef than just top side of the reef.
And the last question we have is regarding the whole conversation about research and sunscreens. They're interested in learning if that's something that you mentioned that research is being done. Is that something that the moat is contributing to?
I believe so. I know in the past that they have touched on it, but I don't think it's necessarily a priority research program for them right now, because the priority is more of the stony coral tissue loss disease right now.
And one last question is, what do you wish we would have asked you today?
I don't know. That's a good question. I can tell you that I wish I didn't flub up in the beginning. But no, I really enjoyed this. This was great. I love being able to talk to people outside of Florida about why it's important to protect our reefs. I hope that you guys can come down and visit us. We do have laboratory tours, and I'm sure, Giovanna, you have my contact information somewhere on the website. If anyone wants to come down, they can contact me, or they can just email me more questions or anything that I can help with. Just let me know.
That would be great. We will definitely add that information to our Reyes website for anyone who's interested in visiting the moat and also learning more. And hey, perhaps next year we can do a live presentation for Reyes from the moat. Wouldn't that be fantastic?
That would be so fantastic. We can even get some activities for you guys to touch some coral.
Well, on behalf of Old Dominion University and the Reyes Program Planning Team, I would like to thank Haley Burleson one more time for joining us today. Thank you, Haley. We will continue to follow your journey and its impact on environmental issues for years to come, because I am sure that you are just getting started, and we cannot wait to continue to follow your extraordinary journey. And to our audience, thank you for tuning in. We look forward to joining us at our next session, which is today at 1 p.m. EDT. where you will learn how to make your own beats. So Haley, if you want to stay with us, you can also learn how to make your own beats. So
Cool.
Don't miss the musical flow of STEM. Today's session with Haley Burleson has been recorded, and if you'd like to watch it again or share it with friends, you can access it from our YouTube channel, which is linked from the Reyes website at odu.edu/Reyes. This concludes our session. Please have a wonderful rest of your day. We look forward to seeing you again here at 1 p.m. EDT. See you soon, everyone. Thanks again.
Thank you.
Bye.